sir.
Okay. Try yourself.
Yes, sir. It's streaming live.
Oh, okay, da. Okay.
Yes, sir.
Okay. We will see. So today, what we are going to discuss, already in our board. So, what is by replica plating method? So, the replica plating method used for the recombinant cells are selected by replica method. That means recombinant cell, how will you select, with the help of the method is called as replica plating method. Okay. The transformed cells are first plate or ampicillin containing mediums. First of all, so some of the medium is there. That medium is containing already, but the agar medium is there. Mostly they were used for the microbial activities, otherwise plant cell or animal cell, whatever it may be, animal cell. Sometimes they were used for the blood and some other components also. Okay. That inactivity medium is contain the specific pH and also the growth hormones and essential amino acids as well as the other substances also present in it. Okay. So here, what they are doing, the recombinant cell, how they will be isolated. After that isolation, what happen, it will grow under the medium. Okay, that medium containing which one, ampicillin containing medium. And after the selection of the transformed form, non-transformed, the colonies are replica plate on medium containing tetracycline for screening of recombinant clones also. So two things is we should note it down here. One samples also grow in a ampicillin containing medium. Another samples also grow under the tetracycline medium. Okay. So both are undergo the screening process. As a result, what happened, those who are resistant to the ampicillins, those who are having the resistant to tetracycline genes, it can be easily identified then only. Okay. See that one. After the incubation of the very viable colonies carrying BP or 322 without DNA insert will appear and position in plate where the non-viable recombinant clones or present can be easily identified. Okay, this is the method. After the incubation period, what will happen? It will what happened? Viability, it can be grow, isn't it? Viable colonies carrying. Which one is alive nature, with which one uptake the nutrition from the agar medium or any other nutritive medium, it will grow. During that growth, what happened, automatically, what happened, viable, it can be easily alive in nature. But that such type of colonies, such type of virus or bacteria, what happened? They carry the BP or 322 without DNA insert. There is no insertion. Okay, only we are growing under the medium only. So that time what happened? Okay. So will appear under the position in plate where the non-viable recombinant clones are present can be easily identified. Which one is moving, which one is non-viable, which one is viable. We can easily identify it during these conditions. Then using the original master plate, these recombinant clones are picked up and subculture. Which one is viable in nature? Already in our board. Tissue culture. Okay, take an examples of any one of the microbial culture. Okay, cereal dilution and also liquid broth culture, and so many culture is that. Okay, subculture means what is the meaning of subculture? So already you know, but this is I see that. See that. So I will show you. Then only it will be better for your understanding. Okay. Take an example. So a small plate is there. Okay, take an example. This is considered a plate. Okay, just imagine. Otherwise, I will do clearly. So then only better for yours. See, this is considered a plate, isn't it? This is considered a small plate. Okay, inside what happened? There is a nutritive medium is there. The nutritive medium is there. Here, what happened? There is a nutritive medium. Okay, so this is considered as agar medium. This is Petri plate method. This is called as Petri plate method. Petri plate. Okay. So there is a lid also there. That is also covered with a lid, already know. Now another category is also there. That is also with the help of the test tubes. Okay. So already in our but the test tube they are having 5 ml, 10 ml, like that it is, isn't it? So here there is a broth is there. So there is a agar here in this portion. In this portion is also the medium is there. That medium is called as what is that? Agar medium. Okay. This is also a agar. Okay. No. Now, what we are doing, what are the samples we are going to grow? For example, take an example yesterday or day, before the class, we saw it. So there is a small plant. Let's say you take any one of the plant sample. Okay, any one of the plant samples, we can cut any portion. So tip of the shoots and roots and whatever it may be. Okay, we can cut and placed inside the medium. Okay. So after that, what happened? No. So inside what happened already in our report, this is containing what is that? There are so many. So nutritive substances are inside. So okay, these are all medium as well as a nutritive substance. Nutritive substances. Okay, nutritive substance. This is it. So this is the red color. I am telling. Okay, red color. There is a nutritive substance. Now what happened? It's also keep it under the which one that is a favorable condition under incubation period. What will happen? After the 24 hours or 48 hours, you can easily observe it. What happened here? It undergoes the modification because the cells also absorb the nutritive substance from the agar. Okay, it absorb it, and also what happened? It will be grow. So as a result, finally what will happen? So you will get the mass of cells. That mass of cell is called as what is callus. The mass of cells like that. Okay, the mass of cells you will obtain within the test tube only. Okay, within the test tubes. So this is the master culture, otherwise called as mother culture. Is there? After you obtain the callus, okay, after you obtain the callus, okay, after you obtain the callus, the callus is nothing but the mass of cells. Okay, mass of cells means each one it has a specific cells. See here, how many cells? So millions of cells, undifferentiated, uni and monoclonal. Monoclonal means all are same in size, same in nature. Okay, this is the structure. Okay. Likewise, from this one, you have to take the selected one. For example, take an example, say any one of the cell like this. Okay. Again, like this, it is replaced into the another test tube. This is the first culture, isn't it? This is considered as a first culture, and second culture also you have to do. Okay, second culture means take an examples, another test tubes, just like this. Okay. Okay, this one is okay, this one is another test tubes. This test tube is called as what is subculture. Okay, this test tube, what is a subculture? Here they already know, but there is a nutritive medium is there. Here also the specific nutritive substance also there. After this callus formation, this callus also transformation takes place from the callus to the another test tubes. Another test tubes. Another test tube means what happened now. So it is transferred from here to here. Okay, here. Okay, just like this. Okay, this culture is called as what is there? Subculture. Okay, subculture. Okay, this is called as subculture. Okay, so don't forget, my dear. So this one is a mother culture, otherwise called as mother. Okay, otherwise called as mother culture. Mother culture. This one is called as subculture. Okay, after the callus formation, it is also, we can do it. Subculture is maximum, there will be serial dilution. You can take an example, say any one of the serial dilution. Test tube is, this is number 1, subculture 1, S1. Okay, otherwise called a sample 1. Likewise, you can make it so many things is that. Okay, you can make it say S1, S2, S3, okay, S4, and S5. Okay, what is that meaning? So after the formation, there is a culture is there similar to that of the callus. Again here to replace to the this one. Okay, again here to here, and then again here. Okay, so why we are doing this one? First of all, you should note it down. So this is the master one, isn't it? This when we are doing this one, the master culture, what happened? There is a callus formation which takes place in this category. Some impurities as well as what is it? It is not a pure one because the first culture, so it is not the pure one. When you are doing the serial dilution methodology, otherwise called a serial, what is a subculture? When you are going to do it, what happened? During this person is 70% is infective manner, means in some impurities, means here say 60%, here 40%, here 20%, here only the 5% of impurities will be obtained. So this final culture callus is also used for the hardening process because we need a 100% pure resistance of the each one. Likewise, what happened, the DNA molecules also, what is the culture under the medium, resistant medium, which one is the tetracycline, ampicillin, like that. Okay, finally, what happened? So when you are doing this one, the plate methodology, okay, so this is the first one, means you can do it, second one, third one, fourth one, up to 10, for example, up to 10. So which one is highly effective, so this part only. 10th one, it is a highly effective one, so it can be used for the further process. So this is the methodology they were used. Okay, my dear, take it out. So this is the things. Say that. So subculture using the same procedure to be obtained pure recombinant clone. That is very, very important. Already told you. Okay. So when you are obtaining pure ones, you have to do more than that. Okay, more than that, the same procedure also. Already what you did in the first procedure, and again it also do the next one. So S2, S3, S4, S5, S6, X7, and up to S10. Okay, you are doing the subculture, the same procedure. What happened? The pure recombinant clone will be obtained in the last one, pure one. Okay, already told you it is highly specific in nature. That is very, very important because we need only pure recombinant clone only is. Okay, we do not know, we do not need which one, impure method, impure 50%, okay, 50%, it is not like that. Okay, pure recombinant clone will be obtained only the subculture only. Okay, so that is the reason that is also doing in this methodology. See that one, for example, say this is the human cells. Okay, gene of the human growth hormone. This is particularly they may be isolated the human growth hormone. Okay, the human growth hormone is also undergoes the cutting process. What is the restriction endonucleases? They have to make a cut any places. After that, what happened? It's also united with the foreign DNA. Okay, so this gene is occupied in this part, and then after that, it also inserted into the E. coli bacteria. Okay, this process. See that one. So this is the one. See, this is also bacterial cell. This is the human cell. They isolate the different type of genomes. Okay. This is the plasmids already in our about. This is the E. coli. So, and also in our about this is the E. coli, another one, animal cell. But here you have to make the cut. Same process also you have to do it. Here the removal is the, this portion is added. So you will get the entire two combination of gene here. So after that, what happened? It's also inserted into the host. Finally, what happened? No, the growth hormone containing genes also, what is a replication process? Okay, it will happen inside the host cell. So finally, what happened? No, these plasmids containing which one? The human genome, human growth hormone. Okay, so that is the important things, my dear. Okay, next one is very, very important, my dear student. This is also another methodology that were used for the marker as well as the insertional recombinant technology. Okay, insertional part. One is called as blue-white screening. Don't forget, my dear, blue-white screening. Blue-white screening. Already we did what is the tetracycline ampicillin screening, isn't it? Here, so they were used the blue-white screening method. Sometimes they were asked the question, what is my blue-white screening? And one more is the why it is also called as chromogenic substrate. What is the meaning of chromogenic substrate? Chrome means color. Genic substrate means the substrate it contain colored substance. Okay. As a result, what it easily visible, because suppose if it is colorless means you cannot able to visible. That's why they were used the color substances. Okay. Okay. The AS markers, don't forget, this is very, very important. Alternative selective marker have also been developed with the differentiate recombinant form, non-recombinant. Recombinant and non-recombinant. How will you identify? Means with the help of the, which one you know? The AS marker. Okay. The AS marker. AS marker means nothing but alternative selectable. Suppose blue means which one is a visible one? White. Black means white. Red means green. Okay. Likewise, your choice. Okay. So the recombinant from non-recombinant on the basis of their ability to produce the color. Suppose some of the microorganisms, they will produce the colored one, isn't it? Potassium dichromate, it is also orange color. Potassium permanganate, violet color. Okay. And take an example, so other things also, pink. Okay, different colors it will be produced. Okay. So that time what happened, you can observe it. So which color it produce. Okay. So it clearly visible means what happened, light color, one dark color. Another one is dark color and light color. Likewise, that is the reasons. The ability to produce the color in the presence of chromogenic substrate. Okay, the chromogenic substrate means, suppose take an example, lactose. Lactose is a white color. Okay, the microorganism also some visible, invisible color means the colorless means what happened, no, it will not easily visible. So the white color substrate is the, when the microorganism act upon it, it produces some other pink color or orange color means what can be, it can be easily visible in nature. That is the reason. That is called as alternative selectable marker. Okay. So when you are using the substrate, depends upon the substrate, they were used the different type of markers. Okay. So in this recombinant DNA is inserted within the coding sequences of the enzyme beta galactosidase. Already you know about it, is also comes under Lac-Operon concept. Lac-Operon concept already know about. There is a structural gene is there. What are the structural genes already you studied, my dear? Dhanalakshmi. Structurally, there are 3 structural genes there. What are they? Do you know?
Beta galactosidase.
No, da. What I'm asking, no, this is the enzyme. Okay. What I'm asking is structural genes. What are the type of structural genes I'm asking? Structural genes. Do you know? Already we did. The things also already we did. Do you understand? See, Lac-Operon concept already you know about, isn't it? Okay. Sorry.
Sir, okay. IAYZ, sir.
Once again.
Sir, ZYA.
Very good.
And I, sir.
Okay. So this one is considered as a Z and Y. All are small a, isn't it? And one more is there, promoter gene, operator gene, and inducer gene, isn't it? Likewise is that. Here, what happened? The Z is produced, which one you know? Beta galactosidase, isn't it? Don't forget, my dear. This is very, very important. Galactosidase, beta galactosidase. Okay, so this, so I will do it. Okay, different colors, then only you understand. Okay, so beta, okay, galactosidase, this one. Okay, galactosidase. Okay, beta galactosidase. Next one is Y gene, isn't it? Y gene is also produced, which one you know? The A gene is also produced, which one? Here, permease. Okay, don't forget, this is very, very important. Permease. Permease. The last one is called as, what is that, you know? Trans acetylase. Don't forget. Okay. Trans acetylase, LAAZ. Okay. Suffix is already in our board. So these are the three combination, three gene is there. ZYS, all are small letter only. Okay. The Z gene produces beta galactosidase. Y gene produces permeases. And then A gene produces trans acetylase. It is very, very important, my dear. Here they have chosen this part only. Z gene produces beta galactosidase. Okay. So that is very, very important, my dear. See here. See that enzyme beta galactosidase. The result into the inactivation of the enzyme, which is referred to as the insertional inactivations. Okay. So in this recombinant DNA is inserted within the coding sequences of the enzyme beta galactosidase. This result into the inactivation of the enzyme, which is referred to as the insertional inactivation. When you are introducing the new genes inside there, what happens automatically that genes are inactivated, okay, by the action of the particular sequence of the enzymes. Okay, so that is very, very important. See then, as a result, what happened? See that the presence of chromogenic substrate gives blue color colonies. If the plasmid in the bacteria does not have an insert, okay, suppose the plasmid in the bacteria does not have an insert, any inserted component, any inserted DNA molecule, it is not present in it, what happened? You know, the bacteria look like a blue in color. The bacteria look like a blue in color without any insertion. Without any insertion, the color will be blue in color. The presence of inserts result. The presence of insert result. What happened into the inactivation of the beta galactosidase and the colony do not produce any color. These are identified as the recombinant colonies. Don't forget, this is very, very important. Okay. Two steps occurs within the culture medium. This is considered as a culture medium. Say that this is a culture medium. Okay. Here you can see it, two types of bacteria. One is called as blue color, another one is white color. It is also depends upon their chromogenic alternative selection. What is that? Alternative selection. See that white patches is also there, white dots, isn't it? Likewise, another one is called as a blue color. Okay, so these are the two things. Then only it can be visible in nature. That's why. Okay, the chromogenic substrate. Okay, see that. See here, the presence of chromogenic substrate gives a blue color colony. If the plasmid is in the bacteria does not have insert, any insertion is not possible. Any insertion is not done in this category. What happened? It look like a which one, you know, blue in color. Suppose if it is inserted, result inserted, okay, inserted any substances. As a result, what happened? The beta galactosidase colonies look like a which color? You know, which color? It will not produce any colors. See, it not produce any color. These are identified as the recombinant colonies because we are inserting. The inserted component is called as recombinant colonies. Okay, so this is the reason. See the next slide, you can easily understand. So this one is blue. Okay, this one is blue, isn't it? These blue colors are identified. Which one, you know? Not inserted any recombinant DNA in this part. Suppose this one is white, isn't it? Those who are seeing the white color, they are all are considered getting noted beta galactosidase inserted genes present in it. So this is the identification of the alternative, okay, forms. The same things also given here also. See that this is also another type. So blue and white screening, alternative screening is that. Okay, so go to the next one. See that one, the plasmid vector. So here you know about the ligation already we did. You have to isolate the two. First of all, you should identify the two colonies. After that, you should isolate the plasmid. After the plasmid, you have to make a cut, and then insertion also done. That is called as ligation process. Then foreign DNA you have to add. Then insert with the lag jet, insert outside the lag gel, and no insert. Okay. So the color will be correspondingly. Suppose these two are inserted. Okay. So, but here insert within lag jet. Within the lag jet inserted inside the plasmids. Here insert outside the lag jet. Outside means outer. Okay. So as a result, what happens? It look like how it will be expressed. This is no. So the already we know without insertion, the color will be blue in color. Outside also in look blue color because it is not incorporated with the plasmids, isn't it? So here inserted with the lag jet means it's look like a white in color because this is a recombinant things. Okay, do you understand that one? Okay, so this is very, very important, my dear students. So many times they were asked the questions, which one is blue color, which one is non white in color. White color is the presence of beta galactosidase and also which one, you know, combined recombinant genes present in it. So others two are blue color means what is it? No recombinant, no inserted genes present in it. That is very, very important. Okay. Tools of the recombinant DNA technology. Another one is very, very important. What are the tools that were used for the DNA technology? First of all, you should note it down. One of the main important role is called as enzymes. Another one is already in our, but without a cloning vector, there is no usage of the enzymes. And the third one is called as competent and coast. Coast is also very, very important. These are the three important tools mostly used for the DNA technology, my dear students. First of all, enzyme. What are the enzymes they were used? You know, there are so many enzymes have been reported. Particularly, they have given as the exonuclease and endonuclease. What is by exonuclease? What is by endonuclease? Endo means inside. Exo means outside. You just note it down. We'll see this remaining things. What is my exonuclease? What is my endonucleases? Okay. Endo inside, exo outside. Okay. Cleaving enzymes. These are all are considered getting known as cleaving enzymes. Exonucleases remove the particular part and also send them out. Normal endonuclease, it's mostly present inside the cell and also used for the cleavage purpose. Restriction endonuclease, it is also present inside. So these are all are considered getting known as cleaving enzymes. Lysing enzyme, another one is called as lysing, only splitting. Okay, scissor, molecular scissor, it will look like. Okay, synthesizing enzyme. Some of the components, some of the enzymes are also used for the synthesizing process. Okay, for example, ligase is also one of the synthesizing process because two units or two plasmids are united with the help of the linking process. Okay, so next one is also the same one, joining enzymes. And alkaline phosphatases, all are having lyasing enzymes, restriction endonucleases, normal endonucleases, exonucleases, joining enzyme, and alkaline phosphate. These are all are comes under the getting known as enzyme. These are all are used for the recombinant DNA technology as a tools. Another one is called as cloning vectors. Already we discussed it. What is by cloning vectors? Without cloning vector, there is no any process in genetic engineering. Okay. Particularly carriers, plasmid, bacteriophage. Okay, plasmids, bacteriophage is mostly used for the cloning vector. Okay, another already you studied a cloning mediator vector, cloning less mediator vector. Already we studied, isn't it? Competent host for the transformation with the recombinant DNA. After the formation of this combination means what is it? Hybridization. After the hybridization, it's also need some technology. Okay, advanced technology. Suppose if it is made of chemicals, calcium chloride. Electroporation is nothing but a small needle used for the DNA load. After the loading, what happens? It reaches the target cell. Ballistic technique, same process. Particle bombardment. Bombardment means what is it? It is nothing but a bomb, explosive. As a result, it will reach the target cell. It is also one of the method, bombard. Gene gun method, already we studied. Gene gun method, already know. It is also loaded a particular microliter, 10 to 220 kilobase loaded and also inserted into the target cells. Micro injections, it is also essential for the micro injection is nothing but one of the process that is nothing. They were used the needle. Okay, so these are all are considered that is known as what is a recombinant DNA technology. Then only you will get the success result. Okay, so otherwise, what happened? I will not get the success result. Suppose any impurities present in the enzymes, what happened? It will not give the proper result. Cloning vectors. Suppose other than these cloning vectors, you choose it. No, the process will be. Suppose if it is very long in period, sometimes what happen? You will not get the good result. Another one is, these are the specific method, chemical and electroporation. These are all very, very important. Then only the recombinant plasmids or genes reach the target cells. That is very, very important. Okay. Okay. Go to the next one. Obtaining the foreign gene product. Obtaining the foreign gene product. In almost all recombinant technologies, the ultimate aim is to produce the desirable protein. Desirable gene. After the production of desirable gene, what will they will do? They also inserted into the any one of the animal or plant, whatever it may be. They incorporated the host DNA. After that, what happened? The mRNA process. mRNA also synthesized. The mRNA produce a particular, what is that? Protein molecule, desired protein. Suppose if you want to produce a hemoglobin protein, they can do a desirable protein only. Suppose take an example is keratin. Suppose immunoglobulin, particularly immunoglobulin G, immunoglobulin M, immunoglobulin A, whatever it may be. Desired protein we can easily produce. Ultimate aim, what is that? Desired protein. Okay. From the genetic engineering or DNA technology or biotechnology, whatever. That process completely called as biotechnology. That engineering process means the genetic engineering. Okay. The foreign gene get expressed under the appropriate conditions. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target proteins, one has to consider producing it in a large scale. That is very, very important. Not in the single one. Okay. Only the production is commercial oriented and also large scale, industrial oriented. Insulin production induction, antibody production industries, sorry. At the same time, immunoglobulin, okay, anti-serum production industries, like that. Okay. So these are all interferon, okay, insulin, and also other polypeptides. Okay. So many things is there. Lactate decogenase is also one of the enzymes. So many things is there. Okay. Plasmids also, some of the markers. Okay. They are all considered getting genetic engineering in large scale. Okay. How they will be? See that one? If any proteins encoding genes is expressed in a heterologous host. Okay. If any protein encoding gene is expressed in a heterologous host, it is called as recombinant protein. Heterologous means, what is the meaning of heterologous? Hetero means two different. What is that? Two host. Two different. It is called as recombinant. Then only recombinance means what is that? A sample, B sample. A comes from animals or plant. Otherwise, what happened? It may be a plant sources itself and also animal sources also maybe. And same species also there. Okay. Then the cell harboring cloned genes of the interest may be grown on a small scale in the laboratory. Okay. If you want to produce millions of what is that? Recombinant protein molecule within the small area, that is called as laboratory. We can do it throughout the world. The vaccinations also prepare in a particular laboratory, isn't it? But they were used for which one? Worldwide. That is the reason. That is the important this. Okay. So go to the next one is restriction enzymes. Okay. Restriction enzymes. Already we studied so many times. What is my restriction enzyme? Endonucleases, exonucleases, how it will be, all these things. Anyhow, we have to screening it once again. Restriction enzymes synthesized by microbes. Only microbes. Don't forget, as a defense mechanism. This is very, very important. Restriction enzymes, so many times they were asked. Restriction enzymes also synthesized only by animal, only by microorganism, only by bacteria. Like that, they may ask the question, only by microbes. Okay. A specific endonuclease which can cleave double-stranded DNA at specific site or recognize sequence in a DNA. The restriction enzymes belong to a large classes of enzymes called as nucleases. Collectively, they are all called as what is that? Nucleases. Why they are all called as nucleases, my dear? They are all act upon only in the DNA, RNA only. That is the reason. That is the reason. That's why they are all are called as, what is that, what is that? That is called as, what is that? Nucleus. Only act upon the nucleic acid only. Okay, so nucleic acid is a substrate, nucleases is an enzyme. So substrate as well as the enzyme. So you will get the enzyme substrate complex. Okay, don't forget, my dear student. Restriction enzyme synthesized by microbes as the defense mechanisms. What is the main use of this one? They only act upon the substrate such as DNA and RNA. Okay, that's why they are all are called as, what is, nucleases. Okay, what is the next one? The restriction endonucleases confer an adaptive advantage on bacteria by digestion of foreign DNA, usually from an invading bacteriophage or bacteriovirus. What is the main functions of the, which one? Restriction endonuclease means, suppose unexpectedly, what happened? What happened? The foreign particles such as a bacteriophage invaded, what happened? Automatically the restriction endonucleases act upon it and completely digest the foreign DNA molecules. That is the process. Suppose if it is not possible, what happened? It will suffered by the phage. So that is the reason. See here, I will show you a small example so I can understand. Okay. Take an example. Okay. This is a bacteria. Okay. This is considered a bacteria, isn't it? This is bacteria. Okay, bacteria, isn't it? Bacteria. Okay. Next one is already in our board. Another one is called as phage. Bacteriophage already in our body, isn't it? Bacteriophage already, very well, but hexago, icosahedral, isn't it? Structure is there. There is a 3-part is the, what are the 3-part? One is called as head, simply called as head. This portion is called as, what is it? This portion is called as neck, otherwise called a collar. This is body part. This is the tail. Okay, this is the tail part. So tail, body part, collar. Inside already in our report. Inside what happened, already, very well about the, which one, so DNA molecule. So this is the nucleic acid, simply we may call it as the DNA. Okay, DNA is the, okay. Here the outermost, the body part already in our, but the outermost part is called as, what is that? Protein part. Protein. Okay, the protein is covered outside. ROTEIN, isn't it? Protein part. Protein. This part is protein part. Otherwise called as capsid. Take an example. Capsid. Capsid or capsomere. Each unit is considered as a capsomere. Okay, capsid. So simply called as capsid or capsomere. Okay, but now what happened? The bacteriophages also affect the bacteria. During that process, what happened? Adhering and pouring, that process also done. I draw here simple diagram only. Okay, you can easily understand. Small dot only I put like this. Okay. Okay, so this one. Imagine 2 virus. Okay. Now what happened? It's completely, this portion completely shrink. During this portion shrinkage means what happened? It's also inserted inside, just like a pouring process. Okay, just like a pouring process. So as a result, what happened? The DNA molecule inside is there. The DNA molecule is also inserted into, which one? Here in this portion, it's also inserted into the bacteria. This one is called as viral. What is that? DNA. Simply viral DNA. Inside already in our, but there is a plasmid is there. The plasmid is also present in it. extra chromosomal body. Okay. But now what happened, when the foreign particle, this is called as foreign particle. Okay. This foreign particle enter inside means, so as a result, automatically the restriction endonuclease act upon it. As a result, it may be completely split the molecules. Okay. Completely split the molecules. Okay. Split the molecules, and what will happen? It may be like this. Okay. It may be like this. See that one? Structure will be like that. Inside there is a molecule is there. That molecule will be like this, isn't it? This structure, okay, this structure will be split like this, isn't it? Completely what happened? Disintegration, otherwise called as lysis. Okay, lysis means lysis, completely simple molecule. So it will not act upon it. That is the reason this work was done by which one? Restriction endonucleases. That is given here. Okay. Don't forget, my dears. Once again, repeat for you. This is a bacteria. Bacteria is affected by the bacteriophage. The bacteria already in our board. Bacteriophage is also already know. The bacteria inside what happened? There is a extra chromosomal body present in it, that is called as plasmid. This is considered as a plasmid. Okay. When the bacteriophage is there, when bacteriophage adhere on the surface of the bacteria, what happened? And also it penetrate the the body part inside the bacteria. As a result, what happened? The nucleic acid of the bacteria, viral DNA is inserted into the bacteria. So after that, what happened? Automatically, what happened? The restriction endonuclease act upon it. As a result, they may be split into simple things. This is one of the method. Suppose take an examples of another one. Okay, about the phagocytosis. Phagocytosis is nothing but one of the process. What is the meaning of phagocytosis, Dhanalakshmi? Do you know that what is my phagocytosis? Do you know, Pa?
So cell eating process.
Very good. Which one is doing? Do you know? Any idea?
No, sir.
You don't know. Okay. Phagocytosis, just take an example. This is consists a cell. Okay. Take an example. This is consists a cell. Okay. But now the foreign particles comes inside. Okay. The foreign particles also coming. So take an example. This one is consists. Sorry, this one is not visible clearly. Okay. You can take in examples. So this one. Okay. So take an example. This one is a foreign particles. Okay, just imagine this virus. So when it come and contact here, what happen? Automatically it invading, the cell wall is invading. Okay, adhere on the surface. After that, what happen? It enter inside. It enter inside. Okay. So as a result, what happened? During this process, when the foreign particle enter, as a result, one of the cell-bound organelle, that is called as lysozyme. Okay, lysosome is also there. Lysosome means one of the cell-bound organelles. Okay, the lysosome, what happened? It lysosome, otherwise called as which one? It is also called as suicidal bag. Why it is called as suicidal bag? When the foreign particles enter inside, during that automatically it act upon it. Okay, act upon it. This is the foreign particle, otherwise called as virus particle, otherwise called as virus gene, whatever it may be, foreign particle, vector, or unknown things, isn't it? Unknown things. Okay. So now what happened here? Just imagine. So lysosome automatically activate upon it. So as a result, what happened? Because the lysosomes secrete so many hydrolytic enzymes, so many hydrolytic. I think it's a 60 to 80 hydrolytic enzymes secreted by the lysosomes, lipase, proteases, and so and so. Okay, so it act upon it. As a result, what happened? These which one? The foreign particles. Which one? Foreign particles split into a simple compound. Simple compound like this. Simple compound like this. So these are the process. Okay, these are the process. So after that completion with the complex molecules of this one is also converted to simple molecules. After that, what are the substances they need? They will absorb it. Remaining things will be how it will be. It sent them out. Here, what happened? See, this is the process. Okay, how it will be? See that one. Here, they send them out. So this is the process. This process is called as what is that? Phagocytosis. Phagocytosis. Phago. Okay. Phagocyto. Okay. Phagocytosis. Otherwise called as what is it? Cell eating. What is it? Cell eating. Cell eating process. Okay, this is considered as a cell eating process because this is a cell. Okay, any one of the foreign particles is considered as a cell. When it enter into the cell, automatically the lysosome act upon it. As a result, what happens? The hydrolytic enzymes also secreted by the lysosome. It act upon it. The complex molecule converted into simple molecule. After that conversion, what happens? It may be eliminated to the outside. So this process is called a cell eating process, otherwise called as phagocytosis. Okay. So that's all about I think this phagocytosis process. Okay. This one. See then one. See, the restriction enzymes confer an adaptive advantage on the bacteria by digestion of foreign DNA, usually from an invading bacteria feeds. The resulting DNA fragments can then be further degraded and destroyed by the exonucleases. The endonucleases, what they will do, inside what happens, the complete degrade the molecules, and also exonuclease, what happens, completely send them out. That is also done by the axons. These enzymes are used to cut DNA in a precise and predictable manner. Okay, so this is the important things. So go to the next one. Do you understand this part, my dear Dhanalakshmi?
Yes, sir.
Okay. So next one, they are extensively useful in gene cloning, DNA amplification, and many recombinant DNA technologies. Maximum the exonuclease and endonuclease mostly involved in the amplification of the DNA technology. That is very, very important, my dear. Okay. These are the two kinds of exonucleases and the endonucleases. These are the endonucleases and the exonucleases. What is that? That there are two types of exonucleases. Simply there are two kinds of nucleases, simply called as nucleases. Okay, simply called as two kinds of nucleases. One is called as exonuclease, another one is called as endonuclease. One is called as exonuclease, another one is called as endonucleases. So what will we do? That is very, very important. Exonuclease, what will we do? Endonuclease is what will we do? This is very, very important. So many times they were asked the question, my dear. Don't forget. Exonuclease is remove nucleotide from the end of the DNA. End of the DNA molecule. That is very, very important. Exonuclease is what happened, remove nucleotide from the end of the DNA molecules. Take an example. Okay, take an example. So this is the DNA molecule. Sorry. So this is the DNA molecule. Okay, already you know our board, the 5' same process there, and 3' is also there. Opposite complementary, there is a 5' here, there is a 3' is there, isn't it? Endonuclease is exon. Exonuclease, what they will do? Remove of the part, any part, it can be removed. Remove by which one, exonuclease. Exo means cut them out. Exo. Yes. Okay, exonuclease. That is the important. Endonuclease means it can you know very about. You can made a cut any direction. Endonucleases. We'll see that one. See, this one is called as exonucleases. This one is called as endonucleases. Endonuclease. Okay, endo cut made inside the DNA molecule. Where? Endonucleases. As a result, how it will be attained? Already we discussed that things also, isn't it? Already so many times we saw it. This is a part. So this is the one part. Another part is what is that? The upper part is here, upper part. The lower part, this one. So this is the lower part. Here already but there is a 5' n. So 3' n. So here 3' and 5'. So this end is called as what is it? Already what is this part? This part already we studied. This part is what is that? Do you know? This end is called as what is that? Sticky end. So here also the same. Okay. So this is the endonuclease activities. This is the exonuclease. Exonuclease means any one of the DNA stand. Okay. The sequence will be removed. Which one is the last sequence will be removed? That is very, very important. I'll also see that one. Okay. See here. The exonuclease remove the nucleotide from the end of the DNA molecules, whereas the endonucleases make a cut a specific position within the DNA molecule. The nucleases enzymes that cut the DNA at a unique sequences is called as restriction endonucleases. They cut the DNA in the non-terminal region. Non-terminal region. That is very, very important. Where they cut is made, the non-terminal regions. Okay. So non-terminal regions. So once again, repeat for you. Exonucleases remove a nucleotide from the end of the DNA molecule, whereas the endonucleases make a cut at a specific position within the DNA. Endonucleases make a cut at a specific position within the DNA. The nucleases enzyme that cuts the DNA at a unique sequence is called as restriction. Unique sequence. That is very, very much. That's why it is called as restriction endonuclease. Restriction. There is a specific restriction is there. Okay. It will not cut any position, any sequence. Okay. So that is the reasons. They cut the DNA in the non-terminal region only, non-terminal region. They are also known as the molecular knives, molecular scissors, restriction enzymes, or molecular scalps. So collectively, they are all called as what is that? Molecular scissor, molecular knives, and restriction enzymes or molecular scalpels. Okay. So collectively, they are known as. Okay, this is very, very important, my dear students. This is very, very non-terminal means what is that meaning? Non-terminal. It is not end with the last one. Okay, it will not end. Any in the middle of the DNA molecule, in the center of the DNA molecule, where there is a polyandromic gene is present in it. That position will be what happened. The restriction endonucleases act upon it, and also it make a cuts. Okay, that is very, very important, my dear. See that one. See the structures. So this is the structure already we saw it. This is the bacteria already know about. Inside there is so many genes also present in it. Here the bacteriophage is adhere on the surface of the bacteria. Then insertion is there. See that foreign DNA molecule, the bacteriophage DNA molecules inserted. The DNA molecules undergoes the activity. So it will also split into this one. See that restriction enzyme cleaves the incoming phages DNA at a recognized site. So this one is split into this part. Other enzymes degrade the phage DNA into the smaller fragments. Again, other enzymes also activate upon it. As a result, what happens? The small fragments, it's also split into small fragment in nature. After that, methyl group at the restriction sequences block the restriction enzyme and protect the bacterial DNA from being cleaved. Okay, this is the host DNA and the bacterial host cells, like that they told. And the last one is the restriction endonuclease were discovered by Harper, Norton, and Smith. Don't forget, my dear student, this is very, very important, my dear. The restriction endonucleases were discovered by Harper, Norton, and Smith. Don't forget, this is so many times they were asked the questions. What is who discovered? Which one, Who discovered this part? The restriction endonuclease was discovered by the name of the scientist called as Harper, Northern, and Smith. That is very, very important, my dear. I think that's all about my today class, my dear. So remaining things we will see on the next classes. Don't forget. Okay, once again, go through it. So I will summarize all those things simply, very fastly. Just imagine. Okay. So blue-white screening is also one of the alternative selectable markers mostly used for the chromogenic substances. They are used for chromogenic substances, particularly enzyme beta-galactosidase. Okay. So already you know about that things also. So here you must note it on which one is inserted. Okay. So that will be look like a colored one. Sorry. So which one is not inserted? It is also a blue in color. Which one is inserted? What happened now? It gives a different colors. Okay, that is already we discussed. And then this is the methodology already know about the white colonies, the blue colonies, and also blue colonies. So her plasmids also connected with the foreign DNA molecule, and also the culture in the particular medium. You will get it. What is that? The white colonies. The white colonies is denoted as the which one? You know, the inserted gene is the beta-galactosidase. So other two are not inserted. Okay, because inserted outside the lag, insert with the lag. Here, no insert. So no insert as well as insert outside the lag is also visible in blue in colors. So inserted only it will be white in colors. Already you know about. Okay. So what are the enzymes involved in the DNA technology? Already know about enzymes, cloning vectors. What are the competent host? Is there cleavaging of enzymes? What are the enzymes? Is the carriers already know plasmid and factor features. Competent and ready in order for transmission with the recombinant DNA technology, chemical and electroporation, biological technique, gene control method, and micro injections. Okay, and the next, how will you obtain the foreign gene product? Foreign gene product only obtained for the desirable protein molecules only in large scale. Okay, in laboratory aspects, already know very about that things also. And another one is what is that? You know, restriction enzymes. Restriction enzyme already know about it synthesized only by the microorganism, and so they are collectively called as only nucleases. Okay, so then why it is called as nuclease? It's only act upon the nucleic acids only. That's why they have given as a name is called as nucleases. Okay, so the nucleases is also divided into two categories according to their activities of the position. One is called as exonuclease, another one is called as endonucleases. Exonuclease means remove the nucleotide from the end of the DNA molecules. End of the DNA molecule. Don't forget. Whereas the endonuclease make a cut at a specific position within the DNA molecule. Within the DNA molecule, what is that? You know, where there is a cut, they made, you know, DNA in a non-terminal region. Non-terminal. It is not the end of the terminal. It's middle of the terminal. Already know about. So they are all called as what is that? You know, molecular knives and molecular scissor restriction enzyme or molecular scalpels. Okay, my dears, don't forget. Please keep on practice. Okay, short period you will get the timetable. I think okay. NEET examination point of view already know about May 7th. I think exam is also going to conduct. Okay, so well prepared. Thank you so much for the participation. Thank you. Bye. Take care. Good night. Dhanalishmi, prepare well. Do well. Okay.
Sir, thank you, sir.
Right, Ra. Bye. Take care. Thank you, ma'am.